NAD+ plays a crucial role in numerous cellular processes, and it goes down with age. While NAD+ precursors like NMN and NR have shown promise in animal studies by boosting NAD+ levels, human trials have yielded mixed results. Interestingly, the reduced form of these precursors, NMNH and NRH, may be more potent NAD+ boosters. In this video, we'll delve into the available data on NMNH. We'll explore its potential benefits, discuss the current evidence, and examine the ongoing research in this area. So what is the difference between NMN and NMNH? It's just the hydrogen on this phosphate group. Here, however, it does change the metabolism in the body, and we will look at what this means in a bit. So why do we care about NMNH and whether it can raise NAD levels? NAD+ is an essential molecule for numerous cellular functions, particularly energy production and DNA repair. Unfortunately, NAD levels decline significantly with age. This decline contributes to various age-related health issues. Scientists have explored using NAD+ precursors like NMN and NR to replenish the declining NAD levels. While animal studies have shown promising results at high doses, human trials have yielded inconsistent outcomes. Furthermore, these increases often occur primarily in the blood NAD levels, with less impact on tissue levels. Translating animal doses to human poses challenges. For instance, a mouse model might require 500 milligrams per kilogram of NMN, which translates to a substantial 3 grams for a human. This raises concerns, as NMN can be converted to nicotinamide, which is toxic to the liver at high doses. Moreover, the traditional approach to extrapolating animal doses to humans for supplements may not be suitable for NMN. Unlike substances like physotin, for instance, that are cleared from the body relatively quickly, NMN influences NAD levels over an extended period. Given these limitations, a crucial question arises. Can we develop a more effective NAD booster than NMN or NR to replicate the beneficial effects observed in animal models? The first question is, does NMNH raise NAD, and is it more effective than NMN? Here is a paper looking at NMNH as an NAD precursor in cell culture and in mice in vivo. The paper was published in March 2021. The main author is Dr. Rieckelt Houtcooper, who presented the results at ARDD 2021. A quick review of the abstract to get an idea of the paper. I'm only going to go through the most relevant parts and not do a deep review. As we just covered, NAD is important in cellular metabolism. We look at this and one other paper, both of which described how they made NMNH. So at the time, there did not appear to be a reliable clinical source of this substance. One of the key results was that NMNH did increase NAD levels much more and faster than NMN in vitro cell study. And the metabolic path from NMNH to NAD was not the same as that for NMN. When given to mice, it causes a rapid and sustained increase in NAD in the blood, as well as in the liver, kidney, muscle, brain, brown fat, and heart, though not white fat, which shows that NMNH increases NAD levels more effectively in mice than NMN and shows promise as an NAD precursor. Let's take a look at the actual results that they saw. These graphs refer to an in vitro, that is, using cell cultures in a petri dish, experiment with mouse liver cells. The graph on the left shows the effect of increasing the concentration of NMN and NMNH and the corresponding NAD levels in the cells. NMNH significantly raised NAD in the cells at a lower concentrations than NMN, 5 micromoles, and reached a plateau at 500 micromoles, at which concentration it had reached a 10-fold increase, where NMN only doubled the NAD even at concentrations of 1 millimole. Not shown in these graphs, in the individual cell types, they reported NAD increasing from 2.5-fold in liver cells to 19-fold in brown fat. The graph on the right shows a single concentration of 500 micromoles and the amount of NAD over time. NMN reached a plateau after only 1 hour, while NMNH continued to increase for 6 hours and then remained stable for 24. The authors then tried a test in mice. The mice were injected with either 250 milligrams per kilogram of NMN or NMNH at time t0 with a booster after 20 hours, and were then sacrificed after 24. As a guide, 250 milligrams per kilogram would be roughly 20 milligrams per kilogram for a human, or about 1.5 grams for a 75 kilogram person. We can see that the NAD levels in the blood increased more than twice as much with NMNH when compared to NMN. They also stayed higher. After 4 hours, the NMN group was back to baseline, while the NMNH group continued to see higher NAD levels. They also look at the NAD levels in specific tissues in the mice. The liver and kidney tissues, which are mostly easily targeted, saw increases in both NMN and NMNH, though the NMNH was significantly higher than NMN. NMNH saw a greater than 5-fold increase in liver and 2-fold in kidney. NMNH also saw significant increases in muscle, the brain, and the heart, tissues which are harder to get to, and saw no significant increase with NMN. And finally, NMNH increased the NAD in brown fat, but not white fat. Here, WAT stands for white adipose tissue. Sequentially using enzymatic inhibitors, the authors investigated the metabolic pathways involved in NMNH becoming NAD. It was first converted to NRH outside the cell and pulled into the cell through the normal transporters for NR. However, when going from NRH to NMNH inside the cell, the enzyme was adenosine kinase rather than the NRKs, which convert NR to NMN. Then it was converted into NAD by the NMN NATs in the same way as NMN. Happy New Year! How are your New Year's resolutions coming along? If you're like us at Modern Healthspan, you're probably prioritizing your well-being this year. And we know that good sleep is absolutely essential for feeling our best. And if I had to pick just one thing to boost my sleep and overall health, it would be magnesium. And not just any magnesium. Specifically, I'm talking about Magnesium Breakthrough by Bioptimizers. Magnesium Breakthrough is formulated with 7 different forms of magnesium, each with its own unique benefits. From improving sleep quality and boosting brain function to supporting a healthy metabolism and managing stress, magnesium plays a role in over 600 biochemical reactions in the body. Even if your New Year's resolutions aren't specifically health-focused, it's tough to achieve any goal without good sleep and effective stress management. So why not give yourself a helping hand? Go to bioptimizers.com/modern and use the promo code MODERN10 to receive 10% off your order. Plus, when you subscribe, you'll unlock exclusive discounts, free gifts, and ensure you have a convenient monthly supply. Wishing you a well-rested and successful 2025. There is one other paper which studied NMNH. I will go through this and then compare some points between the papers. This second paper agrees that NMNH increases NAD, but said that it also suppresses glycolysis and the TCA cycle. This paper came out in 2021 as well, from a little bit after the Hout-Cooper paper, but it did not reference it, so I guess the paper had already been submitted for publication before the previous paper. It was by the Tsinghua University in Beijing. So a quick look at the relevant sections of this paper. As mentioned, they also developed their own internal mechanism for generating NMNH. Like the Hout-Cooper study, it was in vitro with cell lines and in vivo with mice. They showed that NMNH worked better than NMN to raise NAD. They saw an increase in NADH, which inhibited glycolysis and the TCA cycle. Glycolysis is where the cell burns glucose to create energy outside the mitochondria. And then the TCA cycle is the process which completes the respiration of the glucose metabolites inside the mitochondria. So in general, suppressing this would not be a good thing. It also induced cell cycle arrest. That is, it stopped the process of cell division. The papers do agree on the impact of NMNH on NAD levels. So HepG2 are human liver cells, and in vitro, both NMN and NMNH raised NAD, though NMNH was many times more effective. The concentration was 100 micromoles. As a reminder, the Hout-Cooper study used 500 micromoles, which was the one that they found most effective. And in vivo, they used an injection of 340 milligrams per kilogram, which had approximately double the impact of NMN. Just for reference, 340 milligrams per kilogram for a mouse is around 27.6 milligrams per kilogram for a human, or about 2 grams for a 75 kilogram person. The Chinese paper also looked at the metabolism of NMNH. They did not look at how the NMNH got into the cell, but the last step was completed by the NMNATs, and with NMNH, this created NADH. The excess NADH seems to be the cause of the inhibition of glycolysis and the TCA cycle. When Dr. Hout-Cooper presented the results in ARD 2021, he specifically mentioned that he did not look at the impact of NMNH on mitochondrial activity, which would have included the TCA cycle. Time for a short diversion into NAD metabolism and its relationship with NADH. One of the key roles of NAD is as a redox agent. During these reactions, by accepting and donating electrons, it moves between NAD+ and NADH. This is the function that NAD plays as part of the electron transport chain, the system that mitochondria use to make ATP. The ratio of NAD+ to NADH is an important part of the redox state of the cell and normally favors NAD+, meaning that the environment is oxidative. The ratio is also used as a signal by other components of the cell. The ratio moves towards NADH with age, potentially because of the decline in NAD+. So in general, increasing NADH is not good, which brings us to the question, does NMNH increase NADH levels in the cell, and are the results from the Chinese study, particularly the decrease in TCA cycle, because of this increased NADH? These graphs came from the Hout-Cooper paper, where they looked at the NAD metabolites in mouse liver cells. In these cells, NAD was boosted 9.3-fold, while NADH was boosted 2.8-fold, so the NAD-NADH ratio improved. In the talk at ARDD 2021, Dr. Hout-Cooper also addressed the NADH level and said that he was concerned that it would be increased, but did not find that it was so. It seems that the NADH was created, but is then quickly converted into NAD. I asked Dr. Joseph Bauer from the University of Pennsylvania, who we have interviewed about NAD before, about the NADH levels and the slowing of the TCA cycle. Just as a note, he was not involved in any of the research, so he is basing this on the papers. He thought it might be a transient effect, perhaps because the NADH was getting directly into the mitochondria, and so slowing down the TCA cycle. It could do this as the output of the TCA cycle is NADH, which is then used in the electron transport chain. If there was sufficient available, it would make sense that the TCA cycle would be inhibited. To see if this was the explanation, the oxygen consumption of the cell would need to be measured to confirm that the respiration had not dropped. A lot of the data is from cell cultures, so it's not clear whether this inhibition would happen in vivo, and if so, for how long, or whether it would reduce the energy output of the cell. Okay, enough about mice. What about humans? I wanted to see if there were any papers published with NMNH in humans. Here are the results from my PubMed search, looking for NMNH clinical trials in the last 5 years. There are none. Cool. So are there any ongoing trials registered on clinicaltrials.gov? Here, searching for NMNH as the intervention. And again, no results. So if there's any human data, I was not able to find it. However, NMNH has been declared as GRAS, generally regarded as safe in the US. This was self-affirmed. Here is my understanding of what this means. There are two ways of getting a GRAS status for a substance. One is to request it from the FDA by providing the documentation to show that it's safe. The other is to self-affirm it. In this process, a panel of experts reviews the data and declares the substance as being GRAS. This second approach has been taken in the case of NMNH and does not require registration with the FDA. The organization making the statement is Ephephal, and they have called their NMNH Youth Peak. This is the same company who produces Youthever NMN, which is the provider for ProHealth and Do Not Age. In my correspondence with Dr. Bauer, he also commented on the cell cycle arrest with NMNH and NRH, and they both seem to have this effect, at least in cell cultures. Whether a sufficient concentration can be maintained in vivo to have the same outcome is not clear. So not much new data on NMNH, but it does seem to be much more potent at raising NAD than either of the existing forms, NR or NMN. And it also raises NAD levels in tissues, at least in mice, something that NMN has been shown not to do. One other point is that in both of these in vivo studies, NMNH was injected into the mice. Neither was delivered orally. It would have been especially nice to have seen some human data, but per my search, I was not able to find any. I would be very interested to hear from anyone who is trying NMNH or NRH, and what are your experiences so far? How do they compare to NMN or NR? Thank you for your attention, and I wish you all well.